Higher level biology for the IB Diploma program, Pearson book. C3.2 Defense against disease. Our guiding questions here are: how do body systems recognize pathogens and fight infections? And what factors influence the incidence of disease in populations? Pathogens are viruses, bacteria, and other small organisms that can cause disease. Our first defense against pathogens is to prevent them from entering the body. Our skin and mucous membranes act as barriers. Okay, barrier is something that stops something from coming in, right? A block. So that's the skin and the mucous membranes. Mucous membranes is like what's in your mouth. Okay, that's slimy stuff. Skin that's slimy skin is mucous membrane. Okay. When a pathogen is able to enter our body, we have a two-layered immune system that responds to fight the infection. Our innate immune system consists of white blood cells called phagocytes that recognize pathogens as foreign and engulf them by endocytosis, resulting in their digestion. True immunity is built up over time by our adaptive immune system. Okay, so first we have the barrier, the skin and mucous membranes. Okay, then we have this two-layered immune system, which is the first one is the white blood cells, that are the phagocytes, and then that's the innate immune system, and then we have this adaptive immune system. Okay. This component of our immune system chemically recognizes the specific molecules that make up pathogens. These molecules are called antigens. So antigens are part of the pathogens. A type of white blood cell called lymphocytes cooperates in the presence of specific antigens. Okay, so when these little pieces of foreign substances are recognized, lymphocytes are incorporated there, appear there, are made there. This leads to cell cloning of specific lymphocytes to fight off the pathogens that carry the identified antigen. We're going to get each one of these sentences, it's going to get its own little subsection here, where we're going to go into details about what that means. So if you don't understand right now the corporation of lymphocytes, we're going to get into it. One important component of this response is the production of proteins called antibodies. Okay, antibodies are proteins. Remember that. Long-lived memory lymphocytes remain after an infection, providing long-term immunity. There are many factors that influence the incidence of disease in populations. We only began to understand the causes of diseases about 200 years ago. Since then, great advances have been made in disease prevention, especially in the way that human wastes are treated, and sanitary conditions have been improved for water sources and preparing food. So when they talk about waste, they mean like urine and fecal matter, pee and poo. Identification and treatments of viral diseases have been the most difficult to understand, with many viral diseases remaining and spreading in the population. Vaccines are our best protection against viruses, but there are challenges to their acceptance in some human populations. Bacterial diseases have been successfully treated by antibiotics, but their overuse, that means the overuse of antibiotics, is leading to the emergence of antibiotic-resistant strains of bacteria. Okay, so we have a first defense against pathogens, which is our skin and mucous membranes. Then we have an innate immune system response, and then we have an adaptive immune system response. C3.2.1. Infectious diseases are caused by pathogens. Pathogens are the cause of infectious disease. Students should understand that a broad range, that means many different kinds, of disease-causing organisms can infect humans. A disease-causing organism is known as a pathogen. Okay? So some kind of organism, a little creature, that can cause a disease is called a pathogen. Although typically the term is reserved for viruses, bacteria, fungi, and protists. So those are usually the pathogens. Archaea are not known to cause any diseases in humans. There are other kinds of pathogens, but usually we're talking about viruses, bacteria, fungi, and different protists. Students should be aware that careful observation can lead to important progress. For example, careful observations from 19th century epidemics of childbed fever due to an infection after childbirth in Vienna and cholera in London led to breakthroughs in the control of infectious disease. So we're going to look at two case studies about how two scientists, medical scientists or public health scientists, that area of science, were noticing patterns in the population's patterns of disease and health, and how those observations led to a greater understanding of what causes disease and how to prevent it. And one of them is in London and the other is in Vienna. Okay, so we're going to talk about Jon Snow in London, not Game of Thrones Jon Snow, long before that, a different Jon Snow. And we're also going to talk about Semmelweis, Ignaz Semmelweis in Vienna, in Austria. Pathogens are disease-causing organisms. Right? Pathogens are any viruses, bacteria, fungi, and protists that result in disease upon entry into the body. So once they get in, they wreak havoc, therefore we call them pathogens. The vast majority of these types of organisms are thankfully not pathogenic to humans. So most viruses, bacteria, fungi, and protists are not pathogenic. There are some that are only pathogenic to other animals, which explains why cats, dogs, and farm animals are susceptible to their own diseases. Most of these diseases will not transmit to humans, although there are some exceptions. So usually if your cat gets sick with something, it can't make you sick, or vice versa. But there are exceptions to that. Nature of science. Okay, we're going to see this little picture of this community water pump. Right? Meticulous observations can lead to breakthroughs. In 1854, there was a major cholera outbreak. That's a sickness, very dangerous sickness, in a suburb of London, UK. The Vibrio cholerae bacteria that causes cholera is found in the feces of infected individuals. If you don't know the word feces by now, or how it's spelled, it is spelled Faeces. It means poop. But when you're writing your exam, don't write poop or shit, write feces. That's the correct biological term. Okay? It was common practice in the 1800s for residents to empty human waste into areas in front of their homes. So they would poop or pee in these little buckets or chamber pots, as they call them, little ceramic bowls, wooden bowls, whatever they had, and then they would dump it out the windows out front onto the road. The bacteria moved down through the soil and infected a drinking well. So, a long hole in the ground where you pull up the water, a drinking well, that was used by many in the community. A physician, okay, so that's a doctor, by the name of Jon Snow, suspected cholera was being transmitted in water supplies. Okay, so he was a physician, but kind of also a scientist, right? Because he was noticing he kind of figured out, he did some mapping of where cholera outbreaks were happening, and he noticed that they were clustered around certain water pumps, and one in particular. He suspected cholera was being transmitted in the water supplies. He created a map of all known infections and found one particular well had been used by all the infected people. The well was closed and the number of cholera infections fell. The well was closed. I like the passive voice there. Who closed it? How did it close? If I remember correctly, it doesn't say this in the book, so I could be wrong, but if I remember correctly, he broke the pump. He broke off the handle because nobody listened to him. His idea that disease could be spread by water was very radical and not accepted at the time. And people were not willing to just stop using a pump that was their closest source of water and go somewhere else, because that was very annoying and inconvenient, and they didn't understand why, and they didn't want to listen to him. So he broke the pump. The information and map created by Snow formed the basis of modern epidemiology, studies that trace outbreaks of disease. Pathogenic organisms are not inherently evil. It means they don't have an evil nature. They just happen to use human tissues as food and shelter. We're just their homes. Their means of growth and secretions, what they create their waste, however, can cause us harm. We have begun to learn a great deal about pathogenic organisms and have devised treatments, means we've come up with treatments, for both before and after infection. Okay, so treatments to prevent infection and treatments to help get rid of it once we've got it. Improvements in public health policies also help prevent the spread of pathogenic organisms. Throughout most of recorded history, humans had no real knowledge of the presence of pathogens and blamed infectious diseases on factors that now seem almost nonsensical to us, such as miasma, like they thought that it was something called bad air, or evil spirits is also a thing, or imbalances in the fluids of the body. Those were some older ideas. We must always remember that we live in an age where science is providing information not available to us even 1 or 2 generations ago. So now we're going to read about Semmelweis. In the mid-1800s, a physician called Ignaz Semmelweis in Vienna, Austria, began to study a lethal disease commonly known as childbed fever. So women would go into labor and give birth to a baby and then suddenly get a really high fever and die. And that was called childbed fever. So childbed, that's a very old-fashioned term for giving birth. The disease had alarmingly high rates of infection and death in Vienna's maternity wards. Okay, a ward is a place where you stay, kind of like a hospital ward, like a court, or a place where you would spend the night or a few nights. And maternity means mother, so this was like the place where you went to the hospital to give birth. Semmelweis began to note a difference in infection rates between 2 maternity wards. One ward was staffed by midwives, and the other by physicians and medical students. The rate of infection and death on the physician's ward was much higher than the ward staffed by midwives. And that was interesting because physicians were considered to be like more highly educated and more competent. So why were their patients getting childbed fever and dying more than the midwives who didn't have all that education and expertise? So that was kind of a strange pattern that he was noticing, and he was going like, what's going on here? Semmelweis began to narrow down and eliminate specific differences between the 2 wards, besides who staffed them. Okay, so like it can't just be about expertise. Like, if there's something, might have checked out, like, well, is there something in the in the building in those different corridors? Is there something in the air? But it has to do with where they're located. Is there different types of patients in the two different wards? He started going through all the possible differences. The one difference that proved to be consequential, that means the one that was the answer, was that the physicians had often carried out autopsies. That means the dissection of dead bodies. You open up the dead bodies and see either why they died or just learn about their anatomy before helping women in childbirth. Semmelweis postulated, that means that was his idea, what he said and argued for, that the physicians were carrying small particles that caused disease from the autopsies to the pregnant women. Now, this was before we really knew much about what we call now germ theory, which is that there are microscopic pathogens. So this was a crazy idea at the time, that there were somehow invisible particles surrounding there were some sort of invisible particle things on the dead bodies that physicians were somehow contaminated with when they went and somehow got on them, and then when they went to the women giving birth, it somehow caused a fever in them. If you don't know about the concept of pathogens, that's a wild idea, and people did not believe him. He was ostracized, he was criticized, he was kind of considered pretty cuckoo for this idea. But he ordered the physicians to wash their hands with a chlorine solution before treating patients, and there was an immediate improvement in infection rate in the maternity ward. Okay, so sometimes you can prove an idea by trial and error, basically by saying, okay, well, if I'm wrong, then this won't work, so let's try it. And especially in the case of washing your hands with chlorine, which is a disinfectant, it doesn't have, at least in this case, didn't really have any negative consequences. So it's basically we'll try it and we'll see if it works, and then that will prove that I'm right. But this is far oversimplification of how that went down socially and politically for Semmelweis. You can go onto YouTube and see if you can find some fun videos about Jon Snow and Semmelweis. Incredible men. C3.2.2, skin and mucous membranes as the first line of defense. So remember, we talked about 3 lines of defense. First one is skin and mucous membranes. The second is the innate immune response, and then the third one is the adaptive immune response. So we're taking the first one, skin and mucous membranes. The skin acts as both a physical and a chemical barrier to pathogens, but you're not required to draw or label any diagrams of skin. The best way to stay healthy is to prevent pathogens from having the opportunity to cause disease, so stopping them from getting inside you. One way to do this is to try to stay away from sources of infection, and that is why it's still common to isolate people who have highly transmittable diseases, or we also call that quarantine. To quarantine is to isolate someone with a disease so that they don't spread the infection to anyone else. Obviously, it's not possible to isolate yourself from every potential source of infection. Therefore, the human body has evolved some ingenious, that means very clever, ways of making it difficult for pathogens to enter and start an infection. And one of those ingenious ways is your skin. Think of your skin as having 2 primary layers. The underneath layer is called the dermis. That's a bolded word. Write it down. You need to know that and be able to use that word. Okay, in the exam, when you're writing your answers, you need to know the word dermis. And it is very much alive. It contains sweat glands, capillaries, sensory receptors, and dermal cells, which give structure and strength to the skin. The layer on top of this is called the epidermis. You also need to know that word. So you need to know dermis and epidermis. This epidermal layer is constantly being replaced as the underlying dermal cells die and are moved upwards. This layer of mainly dead cells forms a physical barrier against most pathogens because it is not truly alive. As long as our skin remains intact, we are protected from most pathogens that can enter living tissues. Does make you wonder about the process of exfoliation, right? Removing those layers of dead skin cells from your skin. Is that a good idea or not? If having this layer of mainly dead cells is protecting you from pathogens. Something to think about. This is why it is important to clean and cover cuts and abrasions. Abrasion is a scrape, like a surface level scrape. That's an abrasion of the skin when they do occur. Okay, and in this picture, this is a false color scanning electron micrograph, an SEM. Okay, so these are not actually white and pink. Of the mucous membrane lining of the trachea. The trachea is the tube going down to your lungs. The large white cells are called goblet cells, and they secrete mucus. Remember, secrete is basically like to release it. You know, like your saliva glands secrete spit into your mouth. Okay? So the goblet cells secrete mucus, and hair-like, in fact, it is the goblet cells in your mouth that are secreting saliva, which is mucus. Okay, hair-like cilia in pink are also visible. Sometimes I think I'm using an analogy, and it's actually an example of the phenomenon. Pathogens can enter the body at the few locations that are not covered by skin. What is not covered by skin? The cells of mucous membranes produce and secrete a lining of sticky mucus. No, sorry, I missed a sentence there. These entry points are lined with tissue cells that form a mucous membrane. Okay, so the mucous membranes are not considered skin. They're considered mucous membranes. So, there, it's in your mouth. That's not considered skin. That's considered a mucous membrane. And there are other places on the body as well that have mucous membranes. So the cells of the mucous membranes produce and secrete a lining of sticky mucus. This mucus can trap incoming pathogens and so prevent them from reaching cells that they could infect. Some mucous membrane tissue is lined with cilia. Cilia are hair-like extensions capable of a wave-like movement. This movement carries trapped pathogens up and out of the mucus-lined tissues, such as your trachea, right, where you're breathing. Table 1, here it is, here's table 1, shows some common areas that have a mucous membrane. The trachea, the tube that carries air to and from the lungs, nasal passages, tubes that allow air to enter the nose and then the trachea, nasal passages, the urethra, a tube that carries urine from the bladder to the outside, okay. Vagina, the reproductive tract leading from the uterus to the outside. Okay. So according to an article published by the National Institutes of Health, the NIH, bacteria outnumber their human hosts by about 10-to-1 cells. In a typical human adult, bacteria therefore account for about 2% of the human's body mass. Interesting. C3.2.3, blood clotting minimizes blood loss and infection. Sealing of cuts in skin by blood clotting. You should be able to include the release of clotting factors from platelets and the subsequent cascade pathway that results in rapid conversion of fibrinogen. We'll go with fibrinogen. Fibrinogen? Oh, I bet it's fibrinogen. Let's do that. Let's go with fibrinogen. To fibrin by thrombin and trapping of erythrocytes to form a clot. Here's a bunch of new words. Are we ready to know what they mean? Let's find out. When small blood vessels, such as capillaries, arterioles, and venules, those are 3 kinds of small blood vessels. When they are damaged, blood escapes from the closed circulatory system. Often the damaged blood vessels are in the skin, right? You get a cut or an abrasion on your skin, so that damages the blood vessels. And so pathogens are then able to enter the body. Our bodies have evolved a set of responses to create a clot that seals the damaged blood vessels, so preventing excessive blood loss and helping prevent pathogens from entering the body. Probably eat the body too. Anyway, but entering the body. So our body has a way of creating scabs, right? So if you get a cut or scrape, it'll form a scab. That's a blood clot. Circulating in the blood plasma are a variety of molecules called plasma proteins. These proteins serve many purposes, including some that are involved in clotting. 2 of the clotting proteins are prothrombin and, decide how to pronounce this? Fibrinogen. I should probably look this up. These 2 molecules are always present in blood plasma, but remain inactive until called to action by events associated with bleeding. Also circulating in the bloodstream are cell fragments known as platelets. So platelets are cell fragments, small bits of cells. So they're not full cells. Platelets form in the bone marrow, along with red blood cells, which are called erythrocytes. Okay, so red blood cell. Now it's time to learn the proper medical term for red blood cells. It is erythrocytes. And you need to know that word. It's not enough to say red blood cells. You need to know erythrocytes. And white blood cells, leukocytes. Okay, so now it's time to know these big kid words. Okay, leukocytes and erythrocytes. White blood cells is leukocytes. Red blood cells is erythrocytes. But do not remain as entire cells. Instead, one very large cell, platelets, breaks down into many fragments, and each of the fragments becomes a platelet. Okay, so there's an initial cell formed in the bone marrow, and then it's broken down into these fragments, and those fragments are called platelets. Platelets do not have a nucleus, and they have a relatively short cellular lifespan of about 8 to 10 days. Consider what happens when a small blood vessel is damaged. The damaged cells of the blood vessel release chemicals that stimulate platelets to adhere to the damaged area, forming a plug. So you cut yourself, chemical signals are released, and the platelets come and gather and stick. Adhere means stick. And form and create like a plug. The damaged tissue and platelets release chemicals called clotting factors that convert prothrombin to thrombin. So prothrombin is always in the blood, and then when these clotting factors, you need to know that term as well, when these clotting factors are released by damaged tissue and by platelets, they convert prothrombin to thrombin. And thrombin is an active enzyme that catalyzes the conversion of soluble fibrinogen into the relatively insoluble fibrin. Okay, so you've got fibrinogen floating around in the bloodstream because it's soluble, right? But then when you've got these signals being released by the platelets and the damaged tissue, those signals of the damaged tissue release the chemicals called clotting factors. Okay, so they release the clotting factors, and that takes prothrombin, which is floating around, turns it into thrombin, which is an enzyme, And that enzyme takes soluble fibrinogen and converts it into insoluble fibrin. Okay, so it has to come out of solution. It precipitates. The appropriately named fibrin is a fibrous protein. Fibrous is like string, right? That forms a mesh-like network that helps to stabilize the platelet plug. So you've got the platelets gathering, and then you have this fibrin mesh being created to hold it in place. More and more cellular debris becomes trapped in the fibrin mesh, and soon a stable clot has formed, preventing both further blood loss and the entry of pathogens. So you could, maybe in your head you're remembering this as a scab, that a scab prevents blood loss and it prevents pathogens from coming in, but you need to know that it's that it's called a blood clot. Okay. So in this false color SEM, we see the formation of a blood clot. Okay, so the big red structures there, those are erythrocytes, the red blood cells, right? And that big yellow squishy ball looking thing, that's a leukocyte, a white blood cell. These small little whitish yellow ones, those are platelets. Okay, and then this like silly string style stuff all over the place, that's the fibrin. That's what that mesh looks like. It's kind of amazing. C3.2.4, a two-layered immune system, innate and adaptive. Okay, so we've covered the first defense, which is preventing the pathogens from even coming into our body, right, through mucus, which is sticky. So first we have skin, the layers there that stop pathogens from getting in, especially the dead skin layer, right? And then we've got, which is, what do you remember? Epidermis and dermis, the two-layered skin. That's a defense. And the mucous membrane has this sticky mucus that pathogens get stuck in. And then when we do get openings in our skin or in our mucous membrane, we plug up the openings through blood clots. Okay? So now we're moving on from this barrier method here, going on to a two-layered immune system. So what happens when the pathogen gets in? Okay. So you're going to need to know the differences between the innate immune system and the adaptive immune system. Include the idea that the innate system responds to broad categories of pathogen and does not change during an organism's life. Okay, so your innate immune system is the system you were born with, the system that you will die with, and it reacts to kind of like a broad range of things. Whereas the adaptive system responds in a specific way to particular pathogens, and it builds up a memory of pathogens encountered. So the immune response becomes more effective. Students are not required to know any components of the innate immune system other than phagocytes. Okay, so of the innate immune system, you need to know phagocytes. Humans are born with an immune system called the innate immune system. This first layer of the immune system responds to broad categories of pathogens and does not change during a person's lifetime. For example, the innate system would recognize any bacterium as a bacterium rather than a specific species of bacteria. Okay, so it knows that bacteria are not part of, like it knows that a bacteria is a bacteria and not a cell of your body. Okay, it knows that, but it doesn't know anything specific about bacteria and the different types of bacteria. The basis of the innate immune system is the ability to recognize those things that belong in the human body versus those that do not belong. Okay, so the innate immune system, it knows what in your body is you and what isn't, or at least it's supposed to, unless you have an autoimmune disease, in which case it's malfunctioning. In other words, it can recognize and respond to things that are not self. This includes bacteria, viruses, protists, and fungi, and even things such as pollen and dust. I wonder if it can recognize microplastics. That's probably a field of research these days. The molecules of these foreign or not self entities that can trigger an immune response are called antigens. That's a key term. And you know that antigens are part of the foreign things. Okay, so antigens are not something that you create. They are not part of your body. An antigen is a part of like a few molecules of a foreign substance that should not be in your body. The innate immune response involves activation of a group of leukocytes called phagocytes, which are capable of engulfing invading material by endocytosis. You need to know endocytosis. I'm just going to take a minute here to say that there are some foreign things that live in symbiosis with us, such as like the bacteria in our gut, okay, which our body allows to be there. That's a topic for another day, though. Okay, so leukocytes, remember those are white blood cells. Okay, so there is a type of leukocyte called a phagocyte, and it engulfs. That means engulfing is okay, like it takes it in. So it engulfs invading material by endocytosis. So this engulfing system, it's like it actually takes it into the cell. So that's what endocytosis is. It's when a cell pulls something inside of it, inside the cell membrane. So you see this picture here. This is also a false-colored SEM, but it's of a large phagocyte. So the yellow color is the phagocyte. And then the pink rod shapes, those are bacteria. And the phagocyte is recognizing this bacteria as not self and is in the initial stages of endocytosis. So this is the very beginning step of like pulling these bacteria inside the phagocyte. Ugh. What a way to die for those bacteria. Imagine being murdered through endocytosis. The second layer of human immunity is called the adaptive immune response. This portion of our immune response develops over time and only after exposure to specific antigens of specific pathogens. Okay, so an antigen is a specific molecular arrangement of a specific pathogen. The first exposure to a specific antigen leads to a series of cellular events culminating, that means resulting in, molecules and cells that are long-lived and have the ability to defend the body against a specific pathogen. Okay, so this is the creation of cells that have kind of like a memory of the pathogen. The specific long-lived white blood cells that are formed during the first exposure are called memory cells. Okay, that's an actual term, memory cells, and you need to know it and be able to use it. Upon second exposure to the same pathogen, those specific memory cells, the ones that remember the pathogen, can be activated quickly. They can be so effective in fighting a pathogen that a person may not even realize that they were exposed a second time. The adaptive immune response becomes more effective with age as a person becomes exposed to more pathogens. Okay. C3.2.5, the role of phagocytes. Infection control by phagocytes include amoeboid movement. Okay, this is spelled amoeboid, but I'm pretty sure it's pronounced amoeboid because amoeba is a word we know, right? And that's amoeba. So I think in this case, the OE is an E sound. Include amoeboid movement from blood to sites of infection, where phagocytes recognize pathogens, engulf them by endocytosis, and digest them using enzymes from lysosomes. Phagocytes are leukocytes, which are white blood cells, right? They are capable of an action called amoeboid movement. Bolded term. Should be on your flashcards. Cells capable of amoeboid movement can purposefully extend sections of their plasma membrane, followed by their cytoplasm and organelles. Phagocytes use this type of motion to squeeze their way through capillaries so that they can leave and enter the bloodstream in order to move through body tissues. Have you ever seen like an octopus kind of like extend itself through like a really small area? Like, I don't know, that's what this kind of reminds me of. That these cells can do that. Cells capable of amoeboid, they can be like make themselves like kind of send out part of themselves as like a little protrusion and then pop through. So phagocytes use this type of motion to squeeze their way through capillaries so that they can leave and enter the bloodstream in order to move through body tissues. When a phagocyte encounters something in body tissues that contains antigens, remember what's antigens again? It's molecules, like specific molecules on things that shouldn't be there, on pathogens, and thus is not self. It sends out plasma membrane extensions to engulf the foreign body in a process called endocytosis. You can see this in the picture. The foreign body is brought inside the phagocyte where the hydrolytic enzymes of lysosomes, to digest the potential invader. So there are some enzymes that will break it up. This response by phagocytes is nonspecific and is part of the innate immune system. So here you see, in this picture, a portion of the plasma membrane of a phagocyte engulfing a bacterium by endocytosis. Two stages are shown. The bacteria ends up being encased in a vesicle. Remember vesicles from, that's a word that gets used a lot. You can think about it in the neurons. Remember, the neurotransmitters are in vesicles. Okay, well, in this case, a bacterium is put into a vesicle that is later digested by enzymes from one or more lysosomes. C3.2.6, the role of lymphocytes. Lymphocytes as cells in the adaptive immune system that cooperate to produce antibodies. Students should understand that lymphocytes circulate in the blood and are contained in lymph nodes. They should appreciate that an individual has a very large number of B lymphocytes that each make a specific type of antibody. There are many types of leukocyte that contribute to the human immune system. Two major types are B lymphocytes and T lymphocytes. Sometimes their names are shortened to just B cells and T cells. Lymphocytes continuously circulate in the bloodstream, so they're always there, and are also contained within our lymphatic system, especially within lymph nodes. So see in this picture, you can see where the lymph nodes are. We're going to look at the function of B lymphocytes first, and then explore the functions of T lymphocytes. Here's a diagram showing the location of lymph nodes in the neck and head area. B lymphocytes accumulate in lymph nodes. Each lymph node has lymph vessels bringing lymph fluid in and other lymph vessels taking lymph fluid away. The specific leukocytes called B lymphocytes produce protein molecules called antibodies. Antibodies is a bolded term, you should know it. Put it on your flashcards. As part of the adaptive immune system. Okay, so if you have to write all the different aspects of the adaptive immune system, you better have antibodies as part of it. There are many types of B lymphocytes, and each type is able to synthesize, able to create, a specific antibody. Each specific antibody is able to recognize and bind to a specific antigen. So if you have a measles infection, you would produce one type of antibody, and if you contract a virus that gives you influenza, the flu, you would produce another type of antibody. Each type of antibody is different because each type has been produced in response to a different pathogen. Okay? So So the specificity that an antibody has for a certain antigen is not unlike an enzyme's specificity for a certain substrate or a hormone's specificity for a target protein found only on certain cells. Okay, so we've talked about specificity before, right? So each type of B lymphocyte is a biological factory for synthesizing only one type of antibody. Antibodies are Y-shaped proteins. At the end of the branches of the Y is a binding site, just like receptors have binding sites. Right? The binding sites are where an antibody attaches itself to an antigen. Okay, so you've got an antibody, and it's shaped like a Y. It's a Y-shaped protein, and it has a binding site, and that's where it latches on to an antigen, which is part of a pathogen. Because the antigen is a protein on the surface of a pathogen, such as a bacterium, the antibody thus becomes attached to the pathogen. Each of us has many different types of anti-producing B lymphocyte cells, and each can produce only one type of antibody. Antibodies have specific mechanisms for fighting off infection by a pathogen. Any one antibody has two binding sites. If multiple antibodies bind to a cluster of pathogens, a clump is created because each antibody can potentially bind to two different pathogens. The antibody-bound cluster makes it easier for phagocytes to find and engulf the entire clump. Okay, so these little Y-shaped antigens, they've got two binding sites, so they can bind to two different antigens on two individuals of the same pathogen, right? And then this kind of forms a clump, okay? Because if you have two different antibodies, and this one, so here's an antibody and here's an antibody, and these two are both stuck onto the same pathogen, and these two are stuck onto another individual of the same pathogen, like a second bacteria of the same, it's going to hold it all together. And then the creation of this clump is easier for the phagocyte to say, oh, there's something, and take it in. Many viruses attach to the plasma membrane of a body cell. Oh. So now we're talking about the pathogen attaching. So many viruses attach to the plasma membrane of a body cell. The DNA or RNA of the virus is then injected into the cell. This cell then becomes a cellular factory to make more viruses. The protein coat of the virus, called a capsid, often remains on the outer plasma membrane of the cell and can be recognized by antibodies. Multiple antibodies of the same type use their binding sites to attach to proteins of the capsids. This is a way of marking the infected cell to be engulfed later by phagocytes. Okay, so this time the entire cell that has become the virus factory gets engulfed by the phagocytes.